Okay, right. Let's finish up with the excavata and cover all of the rest of the eukaryotes that we'll talk about except for the animals. And so everything from this week on will be about parasites that are not just parasites of animals, but parasites that are themselves animals. All right, cat, you really need to, ow. Okay, there. Yes, he's not a tabby cat. I call him a stabby cat. So we've been talking about excavata. Here's this slide again. Most have flagella, many have a food groove, many have no mitochondria as such. And I need to let somebody in because I just heard the ding dong. Most of them, however, retain. Okay. Most of them, however, retain highly modified mitochondria known as hydrogenosomes that ferment pyruvate to acetate and generate molecular hydrogen. But they don't carry out the whole Krebs cycle. We've talked about the euglenozoa. That includes the trypanosomes. Heterolobosia includes the brain-eating amoebas. The fornicata is what we ended up on Wednesday. That includes giardia, among others. And then preaxostylates and parabasalians include a lot of insect symbionts, not necessarily parasites. In fact, a lot of them are mutualists, notably the protists that live inside termite guts. So I'm not going to go into too much detail about what makes these groups different from each other. The preaxostylata includes mostly a bunch of tiny flagellates. This is one, it's Monocercomonoides. I think at least one of you looking at the cockroach gut contents saw a few little translucent deedly bobs kind of twitching around, very small, a lot smaller than the gregarines or anything else, maybe 10 microns. Those were probably Monocercomonoides or something else that was related to it. This is another one that hangs out in termite guts. This one's called Streptomastix. And termite gut protists have lots of symbioses themselves with bacteria living on them and or in them. And Streptomastix is actually covered in symbiotic bacteria. Working out exactly what all of these symbionts do is tricky work, and I don't think we fully understand what all of them do. In at least some cases, it's the bacteria inside the protists that actually break down the tiny particles of wood. So it's the bacteria in the protist, in the termite, in your house floor that do the work of actually breaking the wood down. As Ogden Nash wrote, the great poet, some primal termite knocked on wood and tasted it and found it good. And that is why your cousin May fell through the parlor floor today. Oh, yeah, the termite tasted wood and found it good because the termite had symbiotic bacteria and symbiotic protists that hosted symbiotic bacteria. And in other cases, we don't really know what the symbiotic bacteria do. There's a cross-section of Streptomastix. On the left, you've got a three-dimensional scanning electron micrograph. On the right, you've got a cross-section, and the central body of the cell is surrounded by these veins. I count 7 of these extensions sticking out, and in between and all around the veins, they're just packed with symbiotic bacteria. And what they're doing there, I don't think anybody quite knows. So yeah, very complicated ecosystem going on in a termite gut. I tell you what, on Monday, I'll bring in some termites and I'll fire up the microscope camera And we'll spend the first part of lecture looking at some of these guys live. We can't really do it in lab. We don't really have time, but it would be a pity to let this course go without looking up termite guts. So that's all I'm going to say about the pre-axis stylates. They're mostly flagellates. The Parabasalia are also mostly flagellates, and these include human pathogens. This one's Trichomonas vaginalis. I used to see this at the Berkeley Free Clinic. Back in the 90s, I was a volunteer medical lab tech, and I've seen all kinds of bodily secretions that most of us are more comfortable not talking about. I used to look at vaginal samples all the time, and every so often we'd spot Trichomonas vaginalis. It's funny, sometimes I'd see it and it wouldn't be associated with symptoms at all. The woman who'd come in for a checkup would not be feeling particularly bad. So it can be a commensal. That said, if you get too many of them, if that delicately balanced ecosystem goes out of balance, you can get an overgrowth of Trichomonas vaginalis, and the symptoms are not lethal, but kind of uncomfortable. Fortunately, there's a treatment. We give people metronidazole to deal with that. Incidentally, there's a close relative called Trichomonas hominis, which lives in the mouths of both sexes, and to the best of my knowledge, that one is a commensal. It doesn't do any particular harm. Okay, you should be able to see there's 3 flagella that are sticking out of the end over on the left side of the picture, and this is convergent with trypanosoma. The 4th flagellum is webbed to the cell body by this undulating membrane, this fold of the cell membrane that makes a sort of fin. And as far as I know, this is convergent evolution, but you can see it pretty clearly here. The picture's been colorized so that the cell body looks orange and the flagella look yellow. And especially on the lower one, you can see a rod-like structure that's protruding from the bottom of the cell. It's called the axostyle. All right, especially important in Korea. Open axostyle. Don't. Okay, sorry. Okay, so T. vaginalis causes, can be a commensal, but can cause disease in humans. T. hominis is pretty much a commensal in human mouths. Trichomonas fetalis causes infection in cattle. It's sexually transmitted, and it can survive freezing, so cows can get it from artificial insemination. And because, unlike Trichomonas vaginalis, which can't do this in people, Trichomonas fetalis can migrate from the vagina into the uterus, and it can cause spontaneous abortion. We've seen a couple of things that can cause spontaneous abortion in cattle. This is one. And most cows these days are bred by artificial insemination. The old days of keeping a bull around just to make the cows happy, most livestock producers don't seem to do that. And this is potentially a threat because it can be transmitted in semen samples. So be very careful about your frozen semen supplier if you're in the cattle biz. There's other parabasalians, lots of them that are symbionts in termite guts. I've seen Trichomonas species in termites. Trichonympha is a lot of fun to watch because it's this blobby thing that always reminds me of Santa Claus because it shakes when it laughs like a bowl full of jelly. It's got this very variable shape. There's a sort of nose cone at one end, and the one at the lower right, you're actually looking straight down on the nose cone. And surrounding that nose cone, you've got large numbers of very long flagella. Kind of up there at the top left, you can see that those two have lots and lots of flagella sticking off. So they look really shaggy. And especially in the one at the lower left, you can see brown wood particles inside the cytoplasm in the posterior half. So these engulf particles of wood, and then it's symbiotic bacteria inside the trichonympha, inside the termite, inside the parlor floor, that do the actual breakdown of the wood. So it's kind of reminded me, if any of you ever sung the song, The Hole in the Bottom of the Sea, right? There's a bacterium in the trichonympha, in the termite, in your house's foundations. There's bacteria in the trichonympha, in the termite, in your house's foundations. Okay, never mind. Right. This is another really weird one. This is another parabasalian called Divescavina. It's got flagella, but they don't really beat. What enables Divescavina to swim is thousands of bacteria that are attached to its surface. Over there in picture B at the right, you can see these very elongated bacteria that flex back and forth and enable Divescavina to swim. This would be a little bit like powering an airplane by gluing a million flies to the wings. Kind of weird, and I don't know if it would work there, but it seems to work fine for Divescavina. There's a related species called Caducea. It's also covered in bacterial symbionts. You can especially see them clearly in B, where they've been stained green. And you can see all those little green dots on the outside of the cell. Those are symbiotic bacteria. They've also got this rod-like structure running down the middle of the cell called the axostyle. And as they swim, the anterior end, the nose cone, if you like, rotates clockwise. This matters because this was a key piece of evidence for the fluid mosaic model of cell membranes. Now, you know what that is. The idea that membranes are made of, right, phospholipids that look like lollipops with two sticks, right? Some of the sticks are bent and some of them are straight, and they form two layers, this lipid bilayer. I'm assuming you remember this from 1440, if nowhere else. And the membrane is not really solid. It's not like a piece of saran wrap or anything like that, or plastic. It's a fluid in two dimensions, kind of like the film of a soap bubble. Well, there was debate as to exactly what cell membranes were, and Caducea provided a key piece of direct evidence. You couldn't have a solid membrane and have part of the cell perpetually rotating. It would eventually twist in two, like, you know, like making a balloon animal or something like that. If you twist a balloon, you end up pinching it off and making, you know, two little bubbles. The only way that you're going to get this kind of rotation without twisting the cell is if the membrane is a two-dimensional fluid. So Caducea was an important piece of evidence for the fluid mosaic model of cell membranes, which is the one that you've learned. One more that I'll mention that shows up in veterinary practice sometimes, Histomonas meleagritis, causes a disease in fowl, chickens and turkeys, and I guess ducks too, called blackhead disease. The protists look like little amoebas, although they do, or they can go through a flagellated stage. You can see a couple of them indicated with the red arrows. They'll penetrate into host tissue and they'll cause lesions, you know, wounds on the intestines, and in severe cases, they can go after the liver. This is the liver of a chicken, I think, that died of Histomonas infection, and all of those craters on the liver are disease spots caused by Histomonas. So yeah, the liver is pretty badly cratered right there. For some reason, infected birds can develop darkening of the skin, which is why this is called blackhead disease. You know, in addition to digestive symptoms and sometimes liver failure, the skin may turn dark in an infected chicken or turkey. Here's the weird thing. Histomonas, this protist, also infects a roundworm. And this is it. This is the head of a nematode called Heterachus gallinarum. And Histomonas not only infects the nematode, it infects the nematode's egg cells, and it can survive for up to 2 years inside the eggs that the nematode lays. So we've seen this before. It's called interovarian transmission. There are some parasites that can do this sort of thing. So a parasitized nematode will lay eggs that contain Histomonas, and if a bird picks up the eggs and swallows any of them, the bird will get infected with Histomonas and also get infected with the nematode Heterachus. So 2 for the price of 1. That's a Heterachus egg, and there's a 4th species that's involved. Earthworms feed on soil, and they'll pick up the eggs of Heterachus, and they can serve as a paratenic host for Heterachus. So if a chicken eats an earthworm that has eaten the eggs of Heterachus, the chicken gets parasitized by Heterachus, and it may also get parasitized by Histomonas, hitching a ride in the eggs of Heterachus, hitching a ride in the earthworm. So dang, yeah, we've got 4 different species involved here in transmitting Histomonas from one chicken to another. I'm imagining you all going, oh, wow, that's cool. Because, you know, it is, right? So here we are back at this slide talking about the supergroups of eukaryotes. We've covered the amoebozoa. That includes entamoeba, the causative agent of amoebic dysentery. We've talked about SAR. There aren't many parasites in the straminopiles or rhizaria, but we did talk about the alveolata, which includes apicomplexa, as well as a few parasites in the ciliates and the dinoflagellates. And we finished with the excavata. The 2 groups that are left are the archaeplastida, which includes the green plants, the green algae, the red algae, and a few protists that are more closely related to them than to anybody else. And the Opisthoconta includes animals and fungi. We are each other's closest relatives. And it also includes protists that share common ancestry with animals and fungi and not with any of these other groups. So Archaeplastida we can get rid of fairly quickly. There are some plants that parasitize other plants. This is one that you can actually see in one of the fields around where I live. This is fairly common in Arkansas. This mass of yellow filaments is actually growing into the shoots of the host plant and parasitizing it, taking its water, taking its phloem, and eventually completely killing it. This is called dodder, genus Cuscuta. And it's one that you could see around here. Some of the prairie wildflowers that we sometimes see in the nature reserve are parasites or hemiparasites. Their roots will grow connections with the roots of other plants and take some of their nutrients or some of their water. So there are land plants that parasitize other plants. None, as far as I know, parasitize animals. you're not likely to wake up and find a tree growing out of your head or anything like that. At least I hope not. This is so cool, I can't let it go. You're looking at a red alga right here, a red seaweed, and you can see towards the center 3 little lumps of brown plant material growing on the red. The lumps are a parasitic red alga called Plocamacolax, and Plocamacolax transfers its own nuclei into the cells of its host, and they replicate and they transform host cells into masses of parasite cells. This is a red algae that thinks it's a virus. It's really cool. It's really weird how the parasite will inject its own nuclei into a host cell, transform the host cell into a parasite cell, and then the parasite cell will divide into masses of parasite cells which produce new parasitic spores. So that's really awesome, and I wish we had more time to cover that. But yeah, parasitic red algae are pretty freaky. The last major eukaryote group we'll talk about is Opisthokonta, which means flagellum at the rear. Opisthokonts, during at least one stage in the life cycle, have a single posterior flagellum. In most animals, this would be the sperm. Many opisthokonts have lost the flagellum completely, notably most of the fungi. Only the earliest branch on the fungus tree still retains the fungi, a group called the Chytridiomycota. They've still got the flagellum. Most fungi have ditched them completely. There's never a flagellated stage in the life cycle of a mushroom, for example. And so Opisthokonta includes the fungi and the animals and some single-celled protists that are more closely related to them than to anybody else. So this is a fun little amoeba that looks like a fried egg. It's called Nuclearia. And I know we put most of the amoebas in the amoebozoa, but this is a case of convergent evolution. Many critters that are amoebas don't belong in the amoebozoa. We've already seen the brain-eating amoebas that are excavates, not amoebozoans. There are some rhizaria that are amoebas, but not in the amoebozoa. And Nuclearia is more closely related to the fungi than anything else. DNA analysis shows that. So this does not go in the amoebozoa. It's an opisthokonta. Ditto for a funny little amoeba that called Capsospora of Chashaki. Of Chashaki. Yeah, Ovchashak would be a Polish last name. Whatever you call it, it's only 5 microns wide, and it's symbiotic in an African freshwater snail, because somebody has to be. It's a close relative of the animal lineage. Not only has it got similar DNA, but integrin proteins, which in you are one of the things that lock cells together in a multicellular body. Capsospora has them too. Although since Capsospora is single-celled, I'm not entirely sure what it's doing with them, but nonetheless, they're there. So this is an opisthokonta that's more closely related to animals than anything else. And then, of course, the fungi include many parasites of animals and plants. Athlete's foot and ringworm and jock itch are things that you may have heard of. There are lots of fungi that are skin parasites that are annoying but not especially dangerous. There are yeasts that can infect humans. There are also yeasts that can be very dangerous if they get deep into your tissues. Fungal infections of the skin aren't that bad, but certain fungi that can penetrate deep into the tissues can cause life-threatening disease. One called histoplasma can grow in your lungs if you breathe in spores, and that can cause severe respiratory illness. There's another one called pneumocystis that can cause severe disease in immunocompromised patients. Pneumocystis urevechii causes a particularly bad form of pneumonia in patients with full-blown AIDS, for example. But we don't have time to cover fungi in this course. This is animal parasitology, and the fungi not being animals, we're going to leave those out. Except there is one group, it's called the Microsporidia, that used to be considered protists, and you'll still find them. Okay, sorry, I'm adding somebody. I want to make sure that she made it here. Right, I hope so. Right, Microsporidia used to be considered parasitic protists, and you'll find them listed in textbooks and reference books as, you know, protist parasites. Once it became possible to sequence their DNA, it turned out that what they are is stripped down fungi. They include organisms with the smallest genomes among the eukaryotes. You have about 3 billion base pairs in your DNA. There is a microsporidian that gets by with less than 1,000th of that, 2.5 million base pairs, coding for something like 1,800 proteins. That's the smallest genome known among eukaryotes. This is a cross-section through an infective spore. The nucleus is down at the lower left. At this stage, there's actually a pair of nuclei. It's called a dikaryon, if you must know. That's down and to the left. And then up and to the right, you can see a whole bunch of circles, kind of making almost a horseshoe shape just under the wall of the cell. What you're looking at is a slice through a coil, right? Imagine taking a, you know, bed spring and slicing it down the middle. What you'd see would be a bunch of, you know, round cross sections through the wires that you cut. That's what you're seeing here. You're seeing a slice through a coiled up structure called a polar filament. And if you ingest an infective spore, the spore will harpoon a host cell with a polar filament, inject its nuclei into the host cell, and then once inside the cell, the nuclei divide and form a multinucleate structure called a sporoplasm, and that will bud off more spores that can go on to infect more cells. So, weird life cycle there. There are 15 species of this that are known to infect humans. Most of them are pretty rare. Most of them are pretty unusual. One was first documented infecting humans in 1985 at the height of the AIDS epidemic, before we had treatments. And this particular one, enterocytozoon, causes wasting and diarrhea in persons with AIDS and other patients that are immunocompromised. It doesn't seem to be too big a problem in healthy humans with functioning immune systems. There are others that infect other vertebrates. There's one called glugia that can infect fish. Sorry, I heard a ding dong. I'm checking to see if anybody. Right, checking to see if I need to add anybody. Seemingly not. There are others that are parasitic in insects. Bees, for example, can get one called nosema that can be rather bad. More recently, they found microsporidian species that infect mosquitoes, and they shorten the mosquitoes' lifespans. At the same time, they also reduce the number of plasmodium that the mosquito may be transmitting. I'm not sure why, and I'm not sure how it works, but mosquitoes that are infected with microsporidians pass on fewer malaria parasites. So this is actively being studied as a means of combating malaria. If you can infect a bunch of mosquitoes, you can cut down on the risk that's entailed by getting bitten by one. So I'm not sure. I don't think the microsporidian actually infects the plasmodium parasites themselves. So I'm not really sure how that works, but that's a potentially useful thing to know, an interesting reason for studying these folks. Okay, by the power vested in me by the state of Arkansas, that's actually all I've got for the slides. So I'm going to go ahead and stop the recording. All right, I thought that would go a little bit longer than it did. Monday, I want to do a little demo of termite gut parasites, and then we'll start talking about parasites that are animals, and that will occupy us for the rest of the course. So I'm going to stop the recording here.